We are back, and yeah, today we'll look at um, some. So we'll looking about these, and for those who are new to the channel, basically here we talk about how can we, how can humans live past 300 years, and yeah, we are covering a wide range of topics, and it's just mainly biology and cell biology and neurons biology. Uh, so last time we were looking at how can we prevent some types of cancer that take place in the brain that affect the neurons. Uh, we were looking at various things that affect the brain cells essentially. So the first thing was that DNA bonds get weak over time because they are being, the DNA is like DNA. It's like this. It's always it's like two strands of DNA and they go intertwining. And then how to pronounce intertwining? Is this even an English word? Intertwining. Oh, yeah, intertwining. Okay, that makes sense.
Intertwining.
Okay, so they go intertwining, and so when basically your genes are made of DNA, your cells in your body are made of this DNA, and then this is what carries the instruction for how your skin color looks like, for everything in your body. And so every single second, it's the mechanism in your body are reading the instructions here. And the instructions basically these two colored things that you see here. These are called a these like adenine, thymine, cytosine, guanine. They are called they are they are nitrogenous bases. I think they are made of a nitrogen group or something. And so these, the way they are coupled, you see like this is this purple is thiamine, this green is adenine, cytosine is this kind of orange, and then this one light blue is guanine. So the way they 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 are sequenced. So like let's say here we have thiamine, and then we have guanine, adenine. So the way they are sequenced is is the information they encode. So basically, when your cell wants to know a protein to code, like let's say we want to code the color scheme or the nail shape or the nails, everything, hair, they lead your DNA. So when you are reading, you have to separate it. The cell have to separate it, like like they separate these two. So let's go on like YouTube and we do DNA transcription video.
Now that we've covered DNA replication, let's talk about transcription. The first thing you need to know is that transcription has nothing to do with cell replication processes such as DNA replication, mitosis, or cell division. So, what is transcription, and why is it necessary?
So here what they were saying is basically cell replication. It's the same thing. I think we separate DNA, but we want to replicate it. So it's like, it's like these two guys. Imagine we cut through here, and then when these two separates, we construct a replica of this, and we add it to the left one, and this which is on the right, we do a replica um, for the left part, and we attach it. Now we have two parts of DNA. That's what we call replication.
So, what is transcription, and why is it necessary? Well, transcription is the first step in the process of using the genetic code in DNA to synthesize or build all the different proteins in your body. One
So that's what I was saying. Transcription is basically when you want to read instructions coded in the DNA to code anything, to code making of enzymes, and enzymes basically are the workforce in your body, like they build up and destroy things. To code, and the enzymes are basically proteins, just like how your nails are proteins. Enzymes are also proteins.
problem with synthesizing these proteins is that the instructions for making them are in the DNA, which is located inside the nucleus.
Yeah, so the DNA is basically inside the nucleus. When you have a cell like this, uh, the nucleus is mostly in the middle, and yeah, we have a bunch of things here like mitochondria. How to spell mitochondria? My English is bad, but we'll be uh, trying together. Okay, no need to waste time here.
Process of using the genetic code in DNA to synthesize or build all the different proteins in your body. One problem with synthesizing these proteins is that the instructions for making them are in the DNA, which is located inside the nucleus. But the place proteins are always made is outside the nucleus, either in the ribosomes floating in the cytoplasm or in the ribosomes embedded in the rough endoplasmic reticulum. So, how does the genetic code for synthesizing?
So yeah, this guy here is rough endoplasmic reticulum, RER. So let's ask this guy. Uh, from evolution perspective. By the way, this color that you see here, it's a small program pro, that I used AI to do, and basically what it is when I'm explaining, I wanted it to be green, and when I'm doing research, I wanted it to be red. So for now, let's make it red so that you know that I'm just also googling things. Uh, because I wanted my viewers to easily know when I'm explaining things or when I'm learning too, so that they can learn with me. So it will be red when I'm just learning, it will be green when I'm explaining. Nucleus. And what is its function? So we just rough endoplasmic reticulum. So just learn that just outside the nucleus you have rough endoplasmic reticulum. Guys, so me when I'm doing biology, I like to know every single thing. Like if I'm watching this video and I'm just watching an hour and that this is a ribosome, but I don't like understand it. What is it? Why is it? We saw that ribosomes are everywhere in the cell.
Well,
Okay,
transcription
like these small guys, these yellow guys are ribosomes. So when
is the first
when
step
I'm
in the
studying, I like to know every single thing. Why
but
is it there? Uh, from evolution, like when the cell was being made, why did the evolution choose that the raphe reticulum is just outside of the nucleus? I like to understand everything that way so that it's easier for me to remember. Like this is one trick, guys. When you are studying, make sure to understand every single thing to be easier for you. So give me a minute, I'm coming. So yeah, that's why I asked this question because you know if you wanna learn something, if you wanna know something, make sure you understand it because understanding it is the first step into making sure that it stays in your brain. Otherwise, if you don't understand it, you are cooked. So yeah, let's understand what this guy does. Okay, so rough endoplazmic reticulum is like is a protein factory. Inside your cell, it's a bunch of flattened sacs. Oh, like pancakes stuck together that are covered in tiny ribosomes. Okay. So the rough endoplasmic reticulum is covered with the ribosomes, and the ribosomes are where protein production takes place. And it's kind of flat, so that you have enough surface area to put enough ribosomes on. I think, uh, yeah. So these are these flat sacs. Yeah, and it's outside the nucleus because we need the DNA to read the instructions to code the proteins, and basically, so yeah, the ribosomes make sense to make them near the nucleus because basically we will copy the DNA instructions into a messenger RNA. A messenger RNA is basically a small part of encoded DNA information uh, that will go and it's basically it resembles a DNA, but it's RNA, but just carries the instruction from DNA into the ribosomes on how to make a protein. Okay. So the mRNA has to be transferred from the nucleus to the ribosome to build protein. It's outside because it needs space to work. Oh, inside the nucleus, that protein protected. Okay. And why is it outside? It's because inside the nucleus basically have no more space. DNA is still packed and protected. Yeah. And the RA just needs room to expand and space to fold proteins. Like when you are making a protein, you need to expand and fold it. So yeah. Okay. So these ribosome can expand and grow bigger maybe when they are making a bigger protein. And you don't want to do that inside of the nucleus where we have our DNA tightly packed and where we basically need a good organization to avoid damage to our DNA. And okay, the rough endoplasmic reticulum is connected directly to the outside of the nucleus membrane. Like I think these guys are very close to the nucleus membrane. Maybe the sacs out here are somehow far, but the other ones are like near. So yeah, that is an advantage because we need to a highway to connect things that are coming from our nucleus to connect the messenger RNA and basically transport it to the ribosomes where transcription is taking place. So yeah.
But the place proteins are always made, RNA polymerase attaches to a genetic code from the synthesizing proteins get from the DNA to the ribosome. DNA uses a messenger called messenger RNA or mRNA to carry the genetic code from the nucleus to the ribosome. The process of building this messenger RNA is called transcription. Now, let's see how transcription happens. Transcription
Okay, so transcription basically is the process of making our messenger RNA that we'll be using in making proteins.
Let's see how transcription happens. Transcription begins when an enzyme called RNA polymerase attaches to a segment of DNA called a gene. A gene
So why is it named RNA polymerase? Why is RNA polymerase named so? Polymerase means it's a polymer. What is a polymer? Oh, so RNA polymerase is basically an enzyme that builds RNA polymers, and the polymer is just a group of monomers. And so DNA is a polymer. So anything made of small, uh, yeah, proteins are also made of amino acids. RNA and DNA are made of nucleotides. Um, Starch is made of glucose and is a polymer. Cellulose is found in plants. Starch is, yeah, it's found in carbs in your food. Like, let's say in corns. And yeah, RNA polymerase now it makes sense. It's an enzyme that builds a polymer of RNA. Nucleotides. And we saw that RNA is made of nucleotides, just the same with DNA. And a nucleotide is basically, do you see like this one guy? And so this yellow and this thing is attached to this is a nucleotide. So these all of them nucleotides. So this is like a monomer, and then the polymer is this chain because there are many. So RNA polymerase is an enzyme that will build a polymer of RNA. Yeah, simple as that.
contains the code to build a specific protein, which is a macromolecule made up of a sequence of amino acids in a specific order. And within a gene, the specific order of nitrogenous bases dictates the order of amino acids that will make up the protein.
Yeah, basically, these nucleotides, these are called um, nitrogenous bases, like the order of these GACT. Uh, they encode information for what protein we will make.
Each group of 3 consecutive bases in the gene is actually a code for a particular amino acid. As a result,
So when we couple these guys 3 by 3, it's a code for an amino acid. So we can ask ourselves, why do we need three? Why do we need three nucleotides to code one amino acid? So an amino acid is basically a monomer for a protein. It's a building block for a protein. It's like a Lego brick for building proteins. And nucleotide is a Lego brick of an RNA. So 3 nucleotides means 3 of these guys. So I'm asking, why do they code just one amino acid? So okay, so now it makes sense. So remember, we are using these to code amino acids that we need to code a protein. And so if we were to use one nucleotide to code for one amino acid, means we will only be able to code for maximum of 4 amino acids, which is not possible because we have total 20 amino acids that can build proteins. So to build a protein, you need Lego bricks. So these Lego bricks to build a protein, they they can be of 20 types. So every single protein in your body is made of combination of 20 amino acids. So to code 20 amino acids, you need codes that can that mean a particular amino acid. So if you have 4 codes, if we decide that one nucleotide codes for one amino acid, it means we would code for maximum of 4 amino acids. If we use 2 nucleotides, it means we would code a maximum of 4 by 4, which is 16, which is not possible. Because you have 4 nucleotides and we are using 2 at a time. So the possible combinations is uh, any of them, 4 of them can link with, you can have AAACAGAU. So A can link with the list of ACGU. Of ACG, is it a U or is it a T? Oh, hmm, GACT. Does we have here a T and there they're saying we need a U? Some use a use nucleotides as letters AUCG and others uh, ACGU. Oh, so DNA has ACGT, but for RNA we have ACGU. Mm, it makes sense now. Okay, now it makes sense. So this is the beauty of biology. Like everything makes sense. Every the, guys, this is why I love biology. Like every single thing have a reason. It's not there by random. It's not there by chance. It's not there to waste our time. Everything is there for a reason. So now we understand why we have. So yeah, back to our topic. We can't use two nucleotides will code for maximum of 16 amino acids, but if you have three nucleotides code for a single amino acid, means we have possible of 64 combinations, which is 4 by 4 by 4. It's like any combination of four nucleotides can combine with another combination of any four nucleotides, and also these can combine with another combination of four nucleotides on their side. So like you can have AAA, CCC, GGG. So now I have 64 different combinations. This is good because it means one amino acid can be coded by actually a group of similar, almost similar nucleotides. So like UUA, UUG, and then here we have CUA, CUG. So you can swap the U with a C and still code for a same amino acid, uh, which is good. So amino acid named leucine can be coded by AUG, and CUG. It means whenever there's a DNA mutation and we end up having a C instead of a U, still would code for the same amino acid. So we are reducing the chances for cancer. Because cancer means basically we are coding the wrong thing. So here, by increasing our chances of not coding the wrong thing, this is good. And also some serve as starting signals and stopping signals to tell our polymerase where to start and end. And our polymerase is that big guy, this guy right here, that codes the RNA.
Each group is referred to as a codon. RNA polymerase.
I don't think my audio is the best because I found that I have to change my output.
Causes a particular area of the DNA helix to unwind and separate into two strands. One of the strands, often called the template strand, is the side of DNA that is read or transcribed by the messenger RNA. The other strand of DNA, often called the non-template strand, isn't transcribed by the messenger RNA. So how are DNA instructions transcribed into messenger RNA? Well, using the template strand as a guide, RNA polymerase uses the base pair rule to assemble free nucleotides in the nucleus into a complementary strand of RNA. For example, RNA polymerase reads the DNA base thymine on the template strand, then binds it to a free nucleotide containing adenine. This process continues with cytosine binding to guanine RNA polymerase
Uracil. Uracil.
Enzyme. Enzyme. Enzyme.
in the nucleus into a complementary strand of RNA. For example, RNA polymerase reads the DNA base thymine on the template strand, then binds it to a free nucleotide containing adenine. This process continues with cytosine binding to guanine and guanine binding with cytosine. With cytosine binding to guanine and guanine binding with cytosine. Remember though, RNA will never contain thymine. So, whenever RNA polymerase sees adenine on the DNA template strand, it pairs adenine with uracil. By using the template strand of DNA as a guide, the genetic code from the non-template strand of DNA has actually been transcribed into messenger RNA. When transcription is complete, the messenger RNA, which is small enough to fit through a nuclear pore, takes the genetic code out of the nucleus to the ribosome, the site of protein synthesis. The process of actually building the protein at the ribosome is called translation, which we'll cover in a separate video. To summarize, transcription is the process of transcribing or copying the genetic code
Oh, my microphone was off this entire time. What? I don't know. So basically, these what about stack premiums? Our HTT uh, is a recruiter. TCNAR is think think of it as a way to fix a damaged small portion of our DNA. And there is also what we call TCNHEG, which can repair when like this whole section is damaged. And so the HTT is involved in the TCNHEG, and basically they create guys that come and fixes this. So increasing these increases our chance of fixing the DNA. Um, so a lesion means faulty code of DNA that been So how does RNA polymerase know that it has to get stuck? And maybe C is paired with not G, but maybe A. So here I'm asking, why in the first place does RNA even get stuck? Like, how does it know that something is faulty? So let's understand it. Oh, okay. So these basically get damaged when there is like another guy attached on the DNA. So because these polymerase needs to read to know if here we have a G, so that we put our C. But sometimes yeah, there can be like another thing attached. I don't know how that happened. Or you know, they are saying that this G can be missing, like there's a space but which is unoccupied. And now this tries to read what is here and can't read it because say that there, but with another thing attached, or it's completely missing. So now it gets stuck. So yeah, now this makes sense. Uh, so yeah, so now we have these two ways to fix the to minimize transcription errors. And remember, we are doing all of these to reduce the chances of cancer or any basically DNA or gene-related issues inside our brain. Because you can't live 300 years if one time maybe we get uh, a small issue with your transcription, and now this builds up and leads maybe to coding of wrong cells, and they get many, and now they cause it. tumor in your brain and now you die. So yeah. Now, so DNA repair is not 100% perfect. Okay. Okay. Now, how can we prevent this? So for each issue, we'll be learning how can we fix it. But most of the things, it's either under trial or never been attempted, or we don't fully understand it yet. But we're here. Our goal is to try to understand the whole concept about everything that is either in our way or that we need to fix or we need to do to achieve basically 300 years of age. Um, so yeah, I don't know if I'm doing the right thing with just doing research. But if you have, I'm planning to like interview some doctors and to give me their, so that they can give me their insights. But for now, what I can do is to even understand what I will ask or to understand what they will tell me. So yeah, that's why I'm spending these hours just trying to understand everything and put everything together, like stitch everything together. Um, so yeah. So now our next question is, tell me the base two clinical trials or active ways to avoid the below issue. Okay, so now for this specific problem of DNA repair not being 100% accurate, we have currently two clinical trials. So the first one is basically lowering the levels of this guy. Yeah. So like the MSH3 is like um, a guy that comes to help repair our DNA. So in some cases, you find that there is repeated CAG many times, and then this guy comes to repair it. But the accident even makes these longer and causes issues. So what they basically do is they inhibit or reduce the volume of these one, and now we reduce the chances of now accidentally even making the problem worse. Uh, and on the other side, this is true from what the research has been showing. Lowering these does not lead to any type of cancer or after effects that can result from the fact that it's now low in volume. So this is the MSH3. So now we prevent these pair problems, but just no, so we're not preventing them. We're just reducing their likelihood. Um, yeah, because they're just reducing the MSH3. So I think here there's no single medicine to cure this. And this method too, basically they are saying preventing the self-destruct signal after DNA damage. But I don't understand why we have to do this while uh, while it's needed. Because when a cell is when a cell is damaged, when DNA is damaged, your body is aware that this cell, if it mutates and becomes a cancerous cell, it will be eating up body resources while basically not contributing to the full body because it's mutated and can't help with anything. So the best solution is the body just lets that cell die because there are billions and billions of cells, and just killing one, it's like a drop in the ocean. Doesn't mean anything. So I don't understand why we have to avoid these to just prevent DNA repair issues. So it doesn't make sense. So yeah, guys, I'm sorry for the background noises. But yeah, we'll continue. Someone is doing some washing of the dishes. So yeah, that's why we have so much noise. Oh, okay, now it makes sense. Okay, now it makes sense. So for the our neurons, we they can't replicate. So if you lose a neuron, if you kill a neuron, you are basically losing it for life because our neurons doesn't do not divide. So for the rest of the body, like let's say for the cells on your skin, you can just kill them when their DNA is damaged because you can make another skin, you can grow another skin, right? But for our neurons in our brain, for the cells in our brain, they don't divide. Now, as a result, instead of just killing it, you can let it become lazy and just kill it. Because imagine you were killing like millions of slightly damaged DNA cells, uh, and now you will end up with reduced mental capability. Because remember, the more neurons you have and the more connections they make, the more your brain understands and processes things. So when you are killing neurons and now the connections they had with each other are now in new sync, and basically the connection between the neurons is our intelligence. It's what makes you remember the color of your car, the house you live in. Now imagine if you're killing those neurons and hence the connection they had are gone. Some memories and some abilities like ability to play a piano or something, you can lose it if you're just killing your neurons. So yeah, it makes sense. For the brain, you don't have to kill. Yeah, you don't have to kill the neurons if they are slightly damaged. And also here, scientists saw that if you don't actually kill it and give it some time, it can heal. Like the DNA repair queue can come and actually fix that particular DNA. So yeah. So now another thing we have about our I'm sorry. Our DNA is because over time as we are opening it up to transcribe it, uh, we are risking some mutations, and each neuron accumulates like 16 mutations per year. But remember, you have billions of neurons in our brain, like 8 billions. Now 16 times billions. Now this is a considerable amount of mutations per year. So what can you do to fix this? I think I have to. I think I have to. For these, I don't think the title is enough. I think for each one of these, we have to maybe summarize it in a sentence or two, um, so that in the future we can know what we are talking about. Okay, now to reduce these mutations, we have seen that first of all, some mutations can come from the fact that we don't have enough nucleotides to build uh, proteins. Oh, to make to make the DNA, and so by increasing the levels of the nucleotides inside our system, we are basically increasing our chances of avoiding the mutations. So this is basically what this is meaning. Because remember, when we are going to do uh, DNA replication, we need nucleotides to build another DNA.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork.
Yeah, so basically when we are going to, to replicate our DNA, we first need to separate it. Uh, and this is done by an enzyme called helicase. And an enzyme
Enzyme.
and an enzyme is a piece of protein that basically does a job, a particular job, if I can say.
The separated strands each provide a temp.
What is the definition of an enzyme?
template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of a new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end. This enzyme makes a small piece. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of
DNA ends are of two types, the 5' and the 3'. Why do we have them, and how to know a 5' end? Okay, so basically these five prime ends is basically a phosphate group, and a phosphate group is these P with five oxygens on the part of a nucleotide. And remember, nucleotide is basically a single strand like these, and where it's attached is a nucleotide. And so it has three parts: there's a pentose sugar, phosphate group, and then nitrogenous base. Nitrogenous base can be the ACGT or something. Um, and then so our phosphate group is the five prime end, and our three prime end is a hydroxide group, the fifth carbon. Okay, I don't fully understand it, but you understand. I wanted to explain this five prime and three prime thing.
Of DNA, the leading strand, is made continuously. The DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Okay, I think that makes sense. But I want to understand why did the evolution prefer having a having a primer enzyme to mark RNA prime on DNA, as where the DNA polymerase should start from when making the DNA. Why not just Okay, so yeah, I was wondering why we need a DNA polymerase and a primer, and I just found that the DNA polymerase can just start. It can't start from scratch. So it's
Adding
like,
bases one by
for
one.
example, it can't
Adding
come
bases one by one.
and start here and start building. It needs to start from another. It has to find an nucleotide to start from to continue. But RNA polymerase can just start from scratch. So yeah. So we need a primer which is an RNA polymerase. Basically that marks like a starting point like here, like we saw in the video, and then the DNA can now can
This
start
marks the
like
starting point for the construct.
so yeah, that makes sense. Guys, I don't know why, but it's just been 1 hour and 30 minutes and I'm feeling so exhausted. I don't know why. I don't know why, honestly. So yeah, for these mutations, so there is two ways. The first one is to basically uh, increase the number of nucleotides that we need to build the DNA, and the other solution is to prevent the DNA from dying. Because remember, mutations can be fixed if you don't just kill the neuron. Rather, the self-fixing can fix actually this mutation because neurons don't divide, they don't replicate, so you can just give it more time instead of just killing it. Let's end today research here. Tomorrow, or if I get courage, I'm feeling exhausted. I don't know why. Because we just covered a few points, but I'm just feeling exhausted. Honestly, I don't know why. But let me take a small pause. I might continue today or tomorrow, but we'll tackle these last 2 points, and then from there we will find other things to work on, like the the surgery will take place maybe to do something on the brain or something to help us achieve 300 years of living. I have a TikTok, but I don't think I've posted anything there because honestly, I think these videos are boring. So there's no need to like clip it and make a TikTok from it. But I just have that account. Thanks for attending today and see you in the next segment.